Here we go.
Good morning. Good evening.
Excellent. As everyone's going to start turning on. Hey, Eric, can I hear you?
Testing. Good. All good.
Oh, good.
Wonderful.
All right. And Dr. Kay is here. Wonderful. I'm going to go ahead and we're going to go ahead and get started. Well, welcome everyone. We're so excited to be here tonight with you. My name is Beth Voss, and I'm the Director of Operations with the Choroideremia Research Foundation. This evening, we're going to be learning about a new company, at least for us, called Kiora. They will be presenting work on the work that they're doing right now on optogenetics on retinitis pigmentosa. Now, I'm going to be glancing at my notes a little bit more tonight because I want to make sure that I hit all the highlights and all the details for all of you tonight. So Kiora Pharmaceuticals is a public technology company focused on developing therapeutics to help patients with orphan retinal diseases. Kiora's lead program, KIO 301, is currently completing its first in-human trial in patients with late-stage retinitis pigmentosa, or RP. Multiple phase 2 trials are currently being prepared for RP, choroideremia, and Stargardt's. KIO 301 is a small molecule that has the potential to restore vision in patients with retinal degenerations. Now, what we want to make sure we do right on, and our guests are certainly going to be talking more about this, the CRF is enthusiastically interested in Kiora's work. And as our presenters will share, at this present time, CRF has not partnered with Kiora, nor are there any participants in any of their clinical trials. There is a memorandum of understanding, an MOU, between Kiora and the CRF that is currently following a grant that they submitted that is currently being reviewed by our Science Advisory Board, which is very exciting. Both Kiora and CRF are exploring the potential of future work together. But as I sit and I listen to this presentation, it brings great anticipation and hope. So that's all good. Now, with all that being said, to clarify our role and expectations of all of you, just a couple quick things. We always want to say thank you to our sponsor, which is 4DMT, for partially funding this evening. There are many people that are joining us tonight. And as I talked with Brian earlier, we're going to take all the questions at the end of the presentation. So if you don't mind, go ahead and use the question and answer or the chat feature, and we'll be sure to get to your questions at the very end. So I think that's it. I want to make sure that I introduce our two presenters, and we do have some people from the CRF joining us tonight, just on board on the sidelines, our executive director, Kathy Wagner, and president of the board, Neil Bench, just in case there's any other future questions for them, but they'll be in the wings. So tonight, who we have with us, we have Mr. Eric Daniels, the Chief Development Officer with Kiora, and also Dr. Christine Kay, who joins us. There she is. And she joins us as well tonight. She is an advising role with Kiora, but she's an international, I want to make sure I say this correctly, vitreoretinal surgeon with lots of international experience with IRDs, and you studied under Dr. Ed Stone, who we certainly are familiar with here at the foundation. He sits on our bench, so we certainly appreciate him. So with all that to say, I think I'm going to go ahead and turn off my video and my screen and turn it over to the two of you, and welcome and thank you.
Well, thank you. This is Eric Daniels from Kiora. As was mentioned, I'm the Chief Development Officer. I think the way we're going to structure tonight's webinar, or this evening, I'm based in Australia, so that's why it's daylight for me. is that we're going to have Dr. K go through really her perspective as a leading clinician in the space and of inherited retinal diseases. And really, this whole relationship kind of was born, probably like many good relationships, out of a beignet and a cup of coffee in New Orleans. Fairly recently. So we have a lot to thank for Dr. K and the team at CRF. And I think that the terms anticipation and hope are great terms to use. So the flow of tonight will be for Dr. K to give, again, her perspective as a clinician on not only on the front line, but on really in the back as well, helping to think people and like such as us to think critically about how we approach drug development. And then I'll spend a bit of time talking about what our drug does, the stages of development, and how we think it could be applicable to CRF. And I'm going to do that through talking about how we currently are seeing it in RP. So with that, Dr. K, I believe you should be able to, as a panelist, share your screen and your slides if they haven't been provided. So if you could just let us know and take it from there.
So I had emailed my PowerPoint to Brian
Okay.
to be able to have him or somebody run the slides.
Yeah. So let me
my
see
update is that I actually lost all power in my house, which is why I'm holding my phone for this Zoom. I have no internet. So even if I wanted to run the slides on my computer right now, I'd have a hard time.
All right, so let me bear with me, and then I will do that.
It's a short PowerPoint, but it would be helpful just to kind of go through, just to keep me on track.
Not a problem, not a problem. So,
Dr. K, are you in Gainesville?
I am in Gainesville. It's ironically not hurricane related. I didn't lose power yesterday. I just literally 10 minutes before, I mean, this is Florida and we have lightning storms all the time. We have a generator. So I do have a full house generator that runs, like, that's why I'm sitting in my room in the light right now. But I couldn't go upstairs where I often do Zoom calls, and my internet goes out. You know, if we get hit by lightning, then internet's down for a little while. But
well, yeah, we appreciate you. We still appreciate you coming on. Thank you so much.
No problem. We were very, very blessed and lucky with the hurricane. We actually only had a mild bit of wind and rain yesterday. So a lot of people in Florida are much worse off than us right now. Okay. So, and just to overview, just to make sure I have this kind of correctly, we want to have about 15 minutes or so for me talking about this. Is
that's about right. Let's do 10 to 15 minutes, and then I'll do the same, and then we'll have Q&A. So
perfect. Okay. So these are my financial disclosures. As we mentioned, I do consult and advise for this company, and for lots of other companies, I'm involved as investigators or as a consultant. So I'm a vitreoretinal surgeon and an IRD specialist, and I'm very actively involved in clinical trials. So I'm a subretinal surgeon in a number of different exciting subretinal gene therapy trials. I do intravitreal injections all day long, shots in the eyes in clinic, either for clinical trials or for my routine clinic. And my passion and interest is inherited retinal disease patients. And again, I, like mentioned before, I studied at the University of Iowa for my fellowship, which is where I really gained an interest and a passion for inherited retinal disease. And see a number of patients with choroideremia. So maybe have met some of you who are on the call today, or some of the families who are on the call today. So we've been living in an exciting last decade or so, really since the approval of Luxturna, which I'm sure you all have heard this word. Luxturna is the FDA approved Retagene and Parvivec gene therapy for subretinal gene therapy for patients with 2 mutations in the RP65 gene, and that was in 2017. And notice we're 6 years from that, and we don't have another yet. And that's been a little bit discouraging. We're just going to talk a little bit about why that might be, and a little bit about the experience of some of these clinical trials. But really, because of what happened with the approval of Luxturna, We really elevated the standard of care in clinical retina to a new standard of care for patients with inherited retinal disease. So it's no longer okay to see a patient with RP or choroideremia and say, there's nothing we can do for you. We don't have any possible therapies. That's not true anymore. So now there are possible, in fact, there's one FDA approved therapy for one disease. It's not choroideremia yet, but it's RP65. And we have, we're going to talk about in a minute, a number of different clinical trials that have either happened or are going to be happening for choroideremia. And so genetic testing is critical, early diagnosis, and then being familiar as a clinical clinician with these clinical trials is critical now as we see these patients in clinic. We can go to the next slide, and that just shows kind of the genetics of, if you go to the next slide, just going to show a little curve of how we now have really increased. This is largely due to Ed Stone and Iowa and other wonderful sites who have really been taking blood samples for decades and trying to identify genes. So look at how that map has gone up and the number of genes that we've identified and mapped for patients with inherited retinal disease. And you can go to the next slide. So choroideremia is an X-linked recessive disease. It's the choroideremia gene encodes for the RAB escort protein, or REP1. It's a rare disease. Somewhere in the range of 1 out of 50,000 patients present with poor night vision or night vision loss in childhood, and this advances as they age. And then they start to notice visual field loss and lose visual field as the decades go by. And the cones, or the central vision cells in the retina, tend to be spared for decades. However, often in the 50s, patients lose their central vision, and patients with choroideremia, end-stage patients with choroideremia, can easily have lost to hand motion, light perception, and that range vision by those decades. This is because of the loss of choroid capillaris, leading to loss of RPE and then photoreceptors. This shows a patient with advanced choroideremia. You can see, interestingly, the most normal area in the retina in this patient is the fovea, right there in the center where it looks red. That actually is what everything should look like. So everything should look red, but you can see everything's white or yellow, and that's with the loss of the normal pigment because of the loss of choroid capillaris and the loss of RPE, which has a melanin dark pigment in it. So everything that's white and yellow is the abnormal area. You can go to the next slide. X-linked recessive, I spelled recessive wrong, sorry, is an important inheritance pattern. It's very important to find the carriers and find the females who are carriers. Women with choroideremia I have a few in my clinic that have an interesting fundus finding. And so we can see some mud splattered fundus and some findings in the females, but tend to not have significant disease, as opposed to other diseases like X-linked RP, where women can be very aggressively affected. I've actually done subretinal gene therapy on one of my females for the RPGR X-linked RP trial, which actually allowed female carriers to be treated. This is not the case for choroideremia. The women tend to be, fortunately, relatively unaffected, except for a few interesting findings in the eyes. Can have some other side effects, but relatively unaffected. No male-to-male transmission since it's on the X chromosome. You can go to the next slide. But it's important for family inheritance understanding and understanding how this is inherited to other family members to identify this X-linked inheritance pattern. Everything in yellow here, these are some of the recent trials for choroideremia. And these, I'm going to go an overview on a few of them. You can just see how many trials were done for choroideremia. These are all subretinal gene therapy trials. And I'll also briefly mention the 4D trial as well today. You can go to the next slide. So it has been a challenging few years since Luxturna. You notice there's not been another FDA-approved gene therapy yet. We have some that are in phase 3 that are exciting. And but the Biogen phase 3, I was an investigator for this, was a huge disappointment to the field for RPGR X-linked RP. It did not quite meet its primary endpoint, which was a visual field endpoint. It got close, but it didn't meet it. And then the Biogen phase 3 for choroideremia STAR program was a big disappointment in the choroideremia field. And I know that I'm preaching to the choir here in the crowd that we have today, but did not meet its primary endpoint, which was a visual acuity endpoint, which we can all argue and talk about whether or not there could have been another endpoint that would have met, or, you know, if we could have done it again, how we would do it. But it's tough sometimes in visual acuity, and the US wants 3 lines of improvement to meet an FDA approval with that endpoint in any disease, but certainly in choroideremia. And then some of the intravitreal trial programs have had some discouraging findings. Some of the inflammation that we're seeing in some of the diabetes program, that was importantly not an inherited retinal disease program, And then the X-linked retinoschisis trial for AGTC, that was an intravitreal, did not show signs of activity. And so some of those trials just were terminated and were not able to show the efficacy or the safety that they wanted. The SEP290 was also a big disappointment, Kiora Therapeutics. And I sent patients to this. I was not a site, but the phase 2/3 did not meet its primary endpoint of visual acuity. You can go to the next slide. So some of this really begs the question, is it the disease? Is it the endpoint? What can we do to improve our interactions with the FDA to be able to look at novel endpoints and new endpoints that might be able to get us across the bar for the threshold of showing efficacy in a meaningful way in patients with low vision with inherited retinal disease, where sometimes the last area affected is the visual acuity. This is a little overview from a paper that I thought was wonderfully done. I think I have the reference on the next slide, but just to go through this, these are some of the choroideremia trials, and it shows that sort of what their visual acuity treatments or gains were in some of these trials for choroideremia. In summary, the 40 patients were enrolled in a number of different subretinal gene therapy trials for choroideremia. Most of them had at least 2 years of follow-up, and there was a median gain of 1.5 letters, which is not enough for an FDA approval, but better than having lost vision with subretinal gene therapy, which is also a possibility. And so this is just a very big landscape overview of some of the choroideremia trials. If you go to the next slide.
Dr. K, actually, if I can just pause for just a second. Why do you think that inflammation remains such an issue in these gene therapy studies? I know that was the previous slide, but can you just comment on that as a general approach and a general limitation, or how we get beyond that?
Yes, and that in itself is like a 2-hour talk, but I'll try to be very brief. First of all, there's subretinal gene therapy, and there's different types of inflammation we see with that. We see deposits in the subretinal space. We used to think the subretinal space was completely immunoprivileged and we weren't going to see any inflammation, but that's not the case. Inflammation is dose dependent. We all saw with the AGTC achromatopsia trial, this is press released and very much out in the public, that there were 3 pediatric dose related serious adverse events with panuveitis and inflammation, very significant subretinal sort of toxicity looking issues with panuveitis. So you can go high enough in the subretinal space with doses that might be too high and cause inflammation. So I think, we've learned even in subretinal gene therapy, you can have inflammation. You need to be careful with your dose, optimize your vector, have a clean capsid design, low empty capsid ratio, and learn from your non-human primate data, what doses we should put in a human eye, and then maybe take a lower dose into pediatrics, because pediatrics, we dose Tylenol, with a lower dose pediatrics. Maybe we should do the same thing with gene therapy. Intravitreal is what the classic, inflammation is that we've all talked about, the Adverum program. And, absolutely, it's a more immunogenic, it's a less immunoprivileged environment. We're not in that safe subretinal space. We're in the vitreous, which we know is more immunogenic. However, we've absolutely learned you can safely deliver various gene therapies into the vitreous, into the intravitreal space. Again, it's still dose dependent. It's also disease dependent. There are some diseases that are just more immunogenic, such as diabetes. We know they have vascular permeability issues, and they're sicker patients, often sort of younger patients too than some of the AMD trials where we're injecting older eyes. So there's a huge long talk there, what the immune system's response is and why it is. And when we see the inflammation with the intravitreal trials, we follow biomarkers like ELISpot and the humoral response, and important things like neutralizing antibodies to see whether or not the immune system is going to attack the AAV vector because it's thinking it's fighting off a virus. And so all these things are really, really important. It's a long talk, but AAV related gene therapy trials, absolutely. We've learned a lot about how to control inflammation. Often we have to pulse these patients with immunosuppression or give them subtenon's Kenalog, which is an injection of steroid at the eye. That can cause risks in glaucoma and cataracts. So it's a very long discussion, but inflammation can be controlled. We have to watch for it, and it usually is more significantly a problem with intravitreal trials than with subretinal, but that's also not always the case. So
that's excellent. Thank you. I'm just trying to give the audience a sort of a general sense of that, and some of the challenges that exist in the field. So
right. So I think in conclusion of subretinal gene therapy trials, BCVA often was the endpoint, and it did show some marginal improvements, but not enough for an FDA approval yet. Surgical technique is important. You know, the choroideremia retina is challenging surgically. It's very thin and fragile. It's actually hard to identify the space because the retina becomes so thin. So it's difficult to even get the cannula subretinally into the subretinal space in a choroideremia eye. And then we need to address when is the right time to treat. So I think we've learned that these trials were really for early patients with choroideremia where we could halt vision loss. You know, maybe if we're very, very lucky and treat at exactly the right time, have some chance for some improvement in maybe visual field in a small area of cells. But this is not restoration therapy. So gene therapy is really treat the viable cells that are left and then halt progression, you know, prevent any loss from there. So let's go to the next slide. The intravitreal trials would be the same goal. This is replacement or augmentation gene therapy to replace the gene. So you have to have viable, healthy cell bodies remaining to target. Patients with LP vision or worse would not be good candidates or would not benefit from these types of trials. And then 4D Molecular Therapeutics has an ongoing, this was from years ago that they announced in 2018, they announced they got orphan drug designation for their vector 4D-110. That's an intravitreally delivered vector with an optimized AAV capsid. And we're waiting to have kind of final results from that trial. It's active, but it's not recruiting at this time. And you can go on to the next slide. So what are the limitations? I already went through that. Requires healthy, viable cells, cannot address advanced stage patients. Intravitreal delivery to be able to actually get through the ILM, which is a membrane that protects the vitreous from getting into the retina. That's challenging. It's hard with an intravitreal vector to be able to transduce, which means to get the vector into the photoreceptors and even deeper into the RPE cells or even the choriocapillaris cells. Which is probably where we need to get for choroideremia. And then subretinal trials also have some risks. You know, it is fragile retina. It's tough and fragile to get into the subretinal space in a patient with choroideremia. It can be done, but we have to be very careful as surgeons in that type of space. Okay, so let's go to the next slide. So this is sort of where the beignet conversation in New Orleans came about. And I think it's interesting. You know, we were talking at Orvo over beignets and coffee and talking about, you know, what about the patients with the low vision? You know, what about that sector of patients? And I actually have a number of patients, and I don't know if any of them are on this call, who are not candidates for any of the clinical trials for choroideremia because their vision was too low. And that's a decent portion of patients in my own population, I'm sure in other IRD clinics. That would like to have something potentially addressable to them. And I think that what we'll talk about tonight is this new potential horizon for patients with low vision with advanced visual loss due to choroideremia as a potential great population of patients to target for some of these novel therapies we'll talk about tonight. And I think the reason the choroideremia is an excellent target for restorative type therapies like we'll talk about tonight is that we understand the disease. We genetically understand the disease. We understand that there's excellent natural history data by virtue of the fact that many clinical trials, as we talked about tonight, have been done. We have an excellent understanding of the natural history of this disease, the progression. We've even had we as the landscape, as the field of IRD specialists, have had multiple conversations with the FDA about good endpoints in this disease, even had advisory panels with the Foundation Fighting Blindness, with the FDA about, you know, this disease and other diseases about good outcome metrics to follow and what would signify an FDA approval to be able to get things across that finish line for an FDA approval. And then maybe most importantly, patients with choroideremia, importantly, did at one point in their life have good historical vision. Now, maybe that is sort of doesn't seem like it's a big point, but it is a huge point. And so that's different than a patient with maybe a very severe loss, severe Leber congenital amaurosis from childhood where they never had good vision and never had established the critical window period time of good cortical development. And so as long as somewhere before the age of 12, patients had that cortical wiring set up with good vision, the brain set up a pathway to have good vision. When that doesn't happen, for instance, a very low vision Leber congenital amaurosis patient from childhood with no light perception vision from childhood, amblyopia. And the word amblyopia means that the brain never learned to see. And it's hard, and we have monkey data from this, and we understand this from humans as well. It's hard, maybe not impossible, but it's very challenging to be able to restore vision if the brain never learned to see before age 12. But that's a wonderful thing about choroideremia, is that it's not an issue. These patients did have good vision at one point in their life and then lost it over time. And as we know, because we're on the phone together, we have a very motivated, organized group on foundations. This is one of the most motivated populations of patients I've had the honor to meet at multiple conferences. I've enjoyed meeting multiple families with choroideremia, and I'm very motivated and inspired by the group of families and the networks that have already been established to raise awareness, raise funding, and work on disease modifying targets for choroideremia. And so I think we can never underestimate the power of group networking and working together, and also working and with groups like the Foundation Fighting Blindness and other nonprofits to help raise money and awareness for diseases like choroideremia and other IRDs. So I think I'll stop there. At least that hopefully opened up the door for what we're going to talk about this evening. And again, thank you for inviting me to be on the Zoom.
It's fantastic. Thanks for really giving people the background and sort of setting the table. Let me ask you just another follow-up question, since I'm going to be speaking about, our drug and the role of RP, because that's what our experience has been. So would you say those two populations that both sit in the ultra low vision state are effectively quite similar, at least from a phenotypic perspective, as how they express their vision loss and sort of the end stage of their disease?
Absolutely. I think that end stage RP and end stage choroideremia often are very difficult. Of course, I have the advantage of being able to get genetic testing very quickly and to know the answer quickly. But if I had a 60-year-old with choroideremia and a 60-year-old with end stage RP come into the room, I might certainly not be able to differentiate them on fundus exam, on vision, on how they even the symptoms that they've told me about, nyctalopia, visual field loss over time. The fundus will look a little bit different, but at age 60, it might actually look fairly similar. And RP retina will start to lose that RPE that will start to become atrophic as well. So I think they're very similar. There are absolutely mutations in RP where the vision loss drops to the level of choroideremia. Not all of them. Some mutations with RP patients will preserve a 20/20 tiny 5-degree island for a very long time, if not forever. It depends on the mutation. So there's a larger mutation spectrum with RP. And so I can't say, all comers with RP are going to end up in that same category. That's absolutely not true. Versus choroideremia is very homogenous. Basically every patient with choroideremia follows a very similar pathway and has a relatively similar disease course.
Perfect. Well, certainly a lot of overlap. So why don't I then switch gears, and that truly serves as the best primer. And bear with me for just a second here.
Hey, Eric, since you're just getting started, maybe I can go ahead and ask a question as you're setting up. Is that okay?
Absolutely.
We have a person. Oh, it just went away. Susie Ford is asking, at what point do you consider a patient in advanced stage? And then I'll hold off the rest of the questions.
I'll let Dr. K answer that one.
So I think specifically in choroideremia, I would say when a patient is lower than is less than meets the Social Security definition of legal blindness might be one way. That might be arbitrary, but worse than 2200. I haven't thought through the answer to this question, so I could be convinced to think about it otherwise. I mean, certainly has to have had some visual acuity decline. So for them to be advanced, they can't be 20/20. They can't probably be 20/40. So must have had some insult to the visual acuity, and so. Which is interesting because in choroideremia, often the vision will, again, age 10, 20, 30, 40, the visual field might be very, very small, but they're often still in that 20/20, 20/40 range, and then until they're not. So then there's this very precipitous decline over a relatively short period of time where the vision can drop from 20/40 and relatively quickly to proceed to 2200, 2400. So I'd say, somewhere in that period of precipitous decline, we're going to call that an advanced stage patient. There are different ways to define that. You could also define it by visual field. So I don't know if I have a great kind of cutoff to say a certain number with visual acuity, but the visual acuity must be worse than 2020.
I'll speak a little bit about that, at least from the clinical trial standpoint, how we define it clinically and from a research perspective. So as was mentioned at the outset, Kiora is a publicly traded company. So forgive me, our lawyers demand that we show you these waiver statements and forward-looking statements. We do focus on orphan retinal diseases. We are equally as passionate because we feel like the overlap between patient needs and physician perspectives like Dr. K's and societies just have great overlaps. And I just feel like that's ultimately, as a company, where we feel like we make the biggest impact. As was discussed, our efforts are in RP, choroideremia and Stargardt's disease. And I'm going to tell you a little bit about RP and our results today. Our efforts and this is really a great relationship to date. It was already decided or elucidated what our relationship is with CRF. We have submitted an application for funding for our additional studies. It is pending. Regardless, it's a collegial relationship to date. A lot of us have a lot to learn from each other's efforts, and so we just look forward to the anticipation and the hope. So let me tell you a little bit about 301. This is a stylized version of your retina. Over time, as Dr. K explained, your retina, your photoreceptors die, but there are cells downstream from your retina, and these are shown in pink, called your retinal ganglion cells. So these are cells that ultimately make up your optic nerve. And what happens, and I'm trying to simplify this really, is that your RGCs remodel when they are not sending or receiving signals from your photoreceptors, which are the cells that are naturally responsible for receiving light. Your retinal ganglion cells change, they modify, and they're looking for signals. They're hungry for those signals that were lost over the progressive decline of your photoreceptors, your rods and your cones. And this is true, as was discussed, not only in choroideremia, but in other inherited retinal diseases like retinitis pigmentosa, which is why I sort of asked Dr. K to set the table. They are very similar when they ultimately get to the sort of later stages. You have degeneration of retina, and what KIO-301 and what our program is about, unlike gene therapies, is, and I want to clarify this from the outset, we are a vision restoration company. Our goal is to restore vision that's been lost. We are not a disease-modifying drug or therapeutic. Now, those two things could be used in concert. We could certainly use a gene therapy for modification or disease modification, and a small molecule like KIO-301 for vision restoration. And I'm going to use that term a lot, small molecule. KIO-301 is a small molecule, and in the world of drug development, you can have things like small molecules, you can have things like proteins, you can have gene therapies, biologics, and as I mentioned, with proteins, the majority of drugs on the planet are small molecules. They are easier to develop. Their pathways are better understood. And so we became attracted to this small molecule approach to treating vision restoration. So now I'm going to really geek a little bit about the science of how these drugs actually work. And at the end of the day, your retinal ganglion cells and all of your neurons, and the retina is really an extension of your brain, if you think about it that way, they all have channels, these ion channels, which control the flow of current back and forth over your membranes and over your cells. And that is what results in signals that go to your brain. And what our molecule does is it's in a class called a photoswitch. And that means that in the presence or absence of light, it changes shape. So you can see here in my slide that when the light is off in your little light bulb here, the KIO-301, it's actually straight, it's linear. And in the presence of light, it bends, it changes shape. It's designed to do that. And what it effectively does is it blocks these ion channels in such a way that it stimulates the cells. So the idea is when light is off, you don't get stimulation. When light is on, you get stimulation of your retinal ganglion cells. So we, if I can go back, are trying to make these pink cells here shown in this illustration photoreceptive or photoresponsive by adding this drug. So the idea again, light on, activate the retinal ganglion cell. That retinal ganglion cell then sends a message to the brain, hey, light is being perceived. So as mentioned, our first foray into this is with RP, and that's why I wanted to set that table. It's very similar, except that it has a very wide range of mutations. I think there are over 150 known mutations, a lot of genetic subtypes. It is an inherited disease. It is an orphan disease. So there are approximately 100,000 patients in the US. And it obviously has a lot of indirect and direct societal costs. Very similar symptomology. You have retention of central vision. It's typically a rod dystrophy first, and then ultimately a cone dystrophy, where folks ultimately lose central vision. We are in the middle of our phase 1B/2 study in Australia, which is where I currently am and live. And we are dosing and treating 6 subjects, and we take them through a series of assessments over a 1-month period after doing an intravitreal injection of drug. So that's an IVT. So Dr. K spoke about a subretinal injection or a subretinal approach versus an intravitreal approach. So intravitreal approaches, I think Dr. K would say without question that from a technical perspective are far easier. And so we're attracted to that. And a lot of groups sort of gloss over, oh, it's a subretinal injection. But as Dr. K noted, it is a very highly technical approach, and one that only in a few hands is likely or certainly possible. Whereas an intravitreal injection, that is the approach that is used to treat diseases like wet AMD. You literally have these injections routinely done throughout the United States and the world on a daily basis. So if you think about the 2 types of patients in this particular study, you want to think about them in 2 buckets. Bucket 1 are patients that are really the sort of worst of the worst. Your no light perception patients or your bare light perception patients. So they range from staring at the sun and not being able to see anything to those that are in the second bucket, who are still able to count fingers or see hand motion, and that's a clinical diagnosis. We do use a specific test to measure their visual acuity, and we don't really need to get into that. But we do the injection and we follow these patients for a month. Then they come back about 60 days later, and we then inject their other eye with a higher dose. And we are in the middle of this study. It is an open label study, which means that we don't have a control. It's not a randomized study. So we're able to really kind of unwrap the Christmas present and look at some of the data along the way. And what we see, I'm going to show you a little bit about what we've seen so far and what our next steps and how it could be applicable to certainly this population. I'll pause for a second, and I think it's helpful for audiences, for folks to know that we have, anytime you do a first-in-man study, really, what are we trying to show? We're trying to show what often is called in our industry as sort of the biological proof of concept. Can you get something and prove the mechanism that's been shown already in animal models? And naturally, can you do that in a safe and tolerable way? So those are the two goals. And I'm not putting weight in one or the other. If I had to, ultimately, it would be safety. You need to prove these are safe and tolerable first and foremost. But we want to prove, is the mechanism possible? Can we reanimate a degenerated retina that's been degenerated for decades? And Dr. K spoke a lot about endpoints, which is it's kind of just a fancy term for assessments and goals, if you will, because when you're developing drugs, you need to be able to prove to others, whether they be regulatory agencies or whether they be clinicians and really our broader community, that what you say is happening is in fact happening, and that not only that's the case, but that those assessments and those endpoints, those goals, are clinically meaningful to somebody. Like, you're making a difference in their life. And that's why, part of that is we want to hear from patients. We want to get their feedback, and you're going to hear from some of our patients in this study. And as I mentioned, safety and tolerability is a must along the way. So again, I'm going to get into some specifics. I can tell you so far that KI-301 has absolutely been safe and well tolerated in our study. And I'm not going to go through every single one of these at what are called adverse events, but we hunt. It is our job and our duty to look for any change in any kind of assessment along the way for any of these patients who are participating in this study. And one of the things that I will point out, this fourth bullet point, is we're not seeing any signs of retinal inflammation. So Dr. K spent a bit of time talking about retinal inflammation and why gene therapies or those approaches can be subject to them. This is where I would say we do have quite a bit of a fork in the road. We are a small molecule. We are less inclined really to kind of generate an immune response in the eye. And the data is supportive of that. So we really don't see any signs of retinal inflammation, and therefore, and we do not dose any of these patients with steroids or as part of a routine treatment. So we, of course, will continue to monitor this and look. But again, so far the safety and tolerability box has been checked, and we're confident. So I mentioned hearing from patients, and really there is no substitute for that. So I do want to spend some time, and I know we're already at 45, and we're probably or at least where I am, it's at that point. But I do think it's important to go through and hear from these subjects who've participated.
Later that week, I went back home to Melbourne, and one of my hobbies is bike riding. And we go out bike riding early in the morning. Often leave when it's first dawn or dark. And as part of that, I put the lights on the bike. And on the tandem bike, I always relied on someone else to say, is the light on? Is it flashing? And because I had no concept myself, and I sort of looked at this light and turned it on and go, hold up, I can see that light, and I can see whether it's on or whether it's flashing. And I think that that was the first time, not only in a clinical sense, but actually it really goes, wow, this is really useful.
Last night when I got home, I was in the city of Kiora yesterday doing some tests, and I've got this device on my bedside, and it's got these two little lights, and I can never see them. And I saw them last night, and I went, wow.
Those are two patients. Sorry, I didn't mention this first subject. He is an NLP patient and has been so for about a decade. Patient 205, her baseline visual acuity is her ability. She can still count fingers. And then you have subject 103, who's a hand motion patient, and you'll hear from him.
It's quite a small area. Again, I thought the light was getting fine to see whether I can see the light. Is it turned on here? And then when I said, I thought, it needs to be made stronger. Well, again, 3 days after the injection, it was like the whole area is struggling. The light was, without me having to look, I could see the light in the ceiling. And it was just like the whole little area is struggling. And I could even see from the kitchen, I could see the glow from that foyer area.
So, look, it's wonderful to hear this feedback and these outcomes and these reports from these patients. It really does mean a lot to us and to our investigators and all the scientists. It keeps us going, among other things, to help continue to drive forward on the progress. So, but of course, we do assess things on an objective measure as well. So we want to hear directly from patients, but we also need to look quantitatively and objectively. Can we see improvement in the visual function of these patients? And we measure that by a handful of different ways. One of them is called your kinetic or your visual field. So we use a device that really measures the degrees of your peripheral vision. And I'm showing you from subject 1-01 what their baseline horizontal and vertical degrees were, and how much improvement that is actually happening at 1 month. So you have about 20 degrees horizontally and about 5 degrees on the vertical side. So again, encouraging signs that we're actually seeing improvements in the degrees of visual field. So and a second measure of how patients do, and this is where one of the things that the Food and Drug Administration likes to see, is they want an assessment of functional vision. So can you represent something in the real world where you would be challenging patients? So we set up and work with partners to set up what's called a high contrast room exit. As you can see, the little schematic here, you have white tiles, and patients need to navigate their way through, and they need to avoid these foam cubes. And we're sneaky. We move the direction and move the location, and it's all random, so you can't memorize where these cubes are. And we also put blindfolds on and occlude one eye. So we put patches on one eye. So we test one eye at a time as well as two. So you can see this is a subject who is a counting finger hand motion patient. This is her baseline ability to navigate through this maze, if you will. So unfortunately for her, we have predetermined grading criteria. And part of why I'm showing this to you as well is to give you a sense of what it would be like to participate in one of these studies. You would definitely be asked to go through something like this. And this is her 28-day or 1 month after receiving the drug. Oops, excuse me. As you can see, much more facile in her ability to navigate through that, quicker. Again, these are all random, so there's no way she's memorizing where those cubes are. This is sort of her tally, if you will. She failed it at baseline, failed it at day 2, and then passed it anytime after that. So these are the things that are encouraging from an objective perspective, showing that, yes, this is supportive further of the type of change that we're making. Also, one of the things we became very interested in doing is we thought, well, we're measuring, we're changing the activity of the retinal ganglion cell. That's what we believe. And the retinal ganglion cell, the tail of the retinal ganglion cell makes up your optic nerve and feeds your brain directly. So we said, well, wouldn't it be fascinating to measure and look at how does the brain react now when you have drug on board or versus baseline? So this is just the setup that we use in the MRI, and we then we present a series of images to these patients that I just showed in the previous slide, and then you look and see, does their brain light up differently? And you can see at baseline, there's not too much activity, and then at day 2, at day 3, a lot of activity shows up right in the middle of the brain, and that is an important segment of what's called your occipital cortex. That's your V1 cortical striate. That is a direct connection to your retina, basically showing that you're getting a lot of retinal activity now when you present and show these images to these patients. And then as you get further out, it doesn't mean that it's not less important real estate. It's incredibly important real estate. You have a lot of vision associated activities that are around there. So we want to see more activity in general. So, but again, an encouraging and also what we call concordant measure, meaning that we're seeing multiple indications, whether we use FMR, whether we use visual acuity, visual field, or whether we use functional tests, that these patients seem to be seeing improvements. So where we stand today is that we are confident so far that the drug is safe and tolerable to date. And We are always probably our toughest skeptics. We do believe that 301 is reanimating the retina, that we have checked that box for biological proof of concept. We've completed enrollment. Doses are ongoing. We'll have this full data set at the end of the year. We are expanding this study based on the initial findings, and I am very happy to share with you and with all of you that we have submitted to the regulatory authorities here in Australia to include an additional 6 patients into this study, which up to 3 of them we are hoping to be end-stage choroideremia patients. So we have submitted, and I'll share with you, we have not heard back, but that process has in fact been initiated. So I want to be very clear about that. And please stay tuned with respect to that, and because we'll have very clear criteria of who would be available or who would be eligible and who would not be. And I promise you that that information will become available public at the appropriate time. We are meeting and regularly do with the Food and Drug Administration to bring our efforts and our studies into the United States, and we'll be following with a second study where we're going to be repeat dosing, not just once a month, or I should say we'll do once a month for 3 months to see that repeat dosing and what that effect that has. And I think that's important because I'm confident there's a learning component here, that when you're restoring light perception or improving somebody's visual field, there's a learning component here. When the brain has, not seen that for what could be decades, that doesn't just happen overnight. And there has to be and is, and other studies have shown this, a relearning of how to interpret that vision and how to make it even more meaningful. We could not do this without the help of our advisory board. As you can see, Dr. K and others. I really do think we have truly a first-class, world-leading group of people that help us think about the things that round out our program. So we try to make the best decisions as possible. I believe you have our contact information, but if not, it's info@kiora.com. And with that, I know we're going over time, but I'm going to stop sharing and take some questions. And I'll let the moderator let us know which questions you want us to field here.
That was awesome. There's so many questions coming in. Do you mind if we just start at the top? And and the two of you can decide who wants to take them. Is that all right?
No problem.
Okay, let's just start at the top. Regarding endpoints, with FDA approved objective visual field testing now available, with requirement from companies like Conan that can be used for non-BCVA based endpoints, what would be the challenges? What would the challenges be for implementing that? It's the first one, if you needed to read that, guys.
Is that where I can actually read it? Because I don't see anything. Am I supposed to be seeing?
Q&A.
What is the term Conan? I'm not familiar with Conan.
I wonder if he means perhaps Kiora. I'm not sure.
Yeah, perhaps.
Should I read that again? Want me to read it again, or is
no,
it?
I can see it with what it says that can be used for non-BCVA based endpoints. So if you click on Q&A, look, I'll let me address that, and then Dr. K, please, because you've been leading the charge.
I'll read that.
And there are no approved endpoints from the Food and Drug Administration for ultra low vision patients today, and so it doesn't mean that it's the Wild Wild West. Far from it. I mean, there are very clear areas where the agency wants to see improvement, and I think there are a lot of incredibly passionate and intelligent clinicians and companies that are working with the agency and advocates working with the agency to define those. I would say it's a shifting landscape, but one that's actually shifting relatively, I don't want to say quickly, but I do think there's movement in that. And I think we will be moving away from sort of legacy methods of getting approval. And I do think the agency is understanding that ultra low vision patients are a unique population, and perhaps we need to change the goalposts, whether that's in kinetic visual field, whether that's in different visual acuity, and whether that's in functional vision. So I don't know, Dr. K, if you want to add or.
I'll say that we as the IRD community are working very much with groups like the Foundation Fighting Blindness to continue to fight the battle of finding good endpoints and defining new novel endpoints and validating endpoints to try to get some endpoints that might be acceptable for pivotal trials. And so, yes, you mentioned visual field. That was actually from glaucoma literature from decades ago, the idea of having 7 decibels increase over 5 grid points. That's a very difficult endpoint. That's why companies like Biogen failed when they tried to take visual field across the finish line, because it's very difficult and you have to repeat it statistically. Visual acuity is also really tough because you have to have 3 lines. I will say there's some positive movement that I've heard, and some of this is sort of hearsay and not at all concrete yet, but that maybe we can help the FDA understand that 3 lines of ETDRS vision is what is accepted now. Maybe we can shift towards 0.3 logMAR in low vision patients and start assigning logMARs to things like no light perception and light perception, because it is life-changing to take a patient from no light perception to light perception. I mean, that should be an approval in my book if I were the FDA, but we need to really work with the FDA and define that. We need to be able to say, okay, we're going to assign this logMAR to, give them numbers and be able to make it defined and statistically significant. And so those types of conversations are sort of slowly happening as well. And then these more novel things like tabletop tests where we move things around and show that a patient has the FDA loves functional vision improvement. And so we're continuing with lots of different IRD communities and the Foundation Fighting Blindness to help define novel endpoints. We know the mobility maze that Spark used to have a patient walk through a room, which is very similar to Kiora's here, but also things like moving objects across the table, and back to what the Argus implant did with kind of using a fork and different tabletop tests. So those are the types of things, getting creative and working with the FDA to be able to show that a patient's function has improved. But it's a slow process, and validating all these endpoints can be difficult, but that's what we all need to do. And the FDA needs to listen. We need to be able to work with the FDA and be able to say, look, we need to find endpoints that we can validate and be able to get some of these therapies up to patients, because that's at the end of the day what we all want to do.
Let's go to the next question. Thank you so much. Linda is asking, as a natural physician, I consider inflammation the body's attempt to heal what's wrong. Why is it considered the enemy in eye disease? Is it possible that it is trying to heal what is wrong in the eye? Thanks, Linda.
Yeah, it's a good question. I'll take that one. And I usually have to explain this to my patients, because you're right, a little bit of inflammation in your arm, not a big deal, goes away. Usually any inflammation in the eye is actually not good. It's something to do with the eye being a small organ, and it can't tolerate inflammation for a long period of time. And that inflammation can start to cause permanent damage. So there's always, too much of something can be bad, so inflammation is definitely really, for example, inflammation, if it's involving multiple layers of an eye, for instance, the optic nerve or the retinal vessels, that can cause almost immediate permanent damage. And so it depends on what layers of the eye are involved. There's minimal inflammation, like a little bit of anterior chamber cell, that's okay. We can treat you with some topical steroids and that can go away. And then we get sort of farther back into the deeper layers of the eye, retinal inflammation, vascular inflammation, optic nerve inflammation, that we don't tolerate. You can go blind overnight with that, and irreparable permanent damage. So it depends on what structure of the eye is involved. But usually speaking, we don't want any inflammation in the eye. It's such a small organ, and these structures are so close to the structures in the back that can't tolerate any inflammation. We want to nip it in the bud and prevent inflammation.
It's a thorough answer. Thank you. Rich wants to know, does affecting or activating the pink neurons in any way conflict with any remaining healthy retina?
Yeah, fantastic question, Rich. I failed to mention one of the things that is unique about KIO 301 as a photo switch is that it is only taken up by pink cells, or in this case, retinal ganglion cells, those retinal ganglion cells that sit downstream from degenerated photoreceptors, from degenerated rods and cones. So it upregulates a certain protein channel that allows the drug in. So if you have normal retina that's remaining, you will not have drug being taken up by those retinal ganglion cells that are still receiving signals. We've done some really, I think, definitive non-clinical animal work showing that 301 really does not affect what we call wild type retina. So the answer to your question is no, it does not conflict with any remaining healthy retina. But that's a fantastic question, because you're absolutely right. We do not want to interfere with normal native vision.
Are all of the patients treated at the lowest dose?
No. So, I mean, in our current study, it is a dose escalating, if you will, study. So we started at 7.5 micrograms, and then we have a group of patients that are treated at 25 micrograms, and then a group of patients, as we're finishing up here, that are treated at 50 micrograms. So we'll continue to monitor the dose. We think what we have is a fairly wide, what we call therapeutic window. And so that's a balance between what we want to see and what we hope to see from an efficacy standpoint, but again, entirely driven by safety. So we haven't seen a safety signal yet, so we'll take the appropriate, conservative and, approaches to dose escalating. With this, not this study is already designed, but that will also be the case in any follow-on study that we do.
I think that in part answers the next question, which Ben asks, will you be increasing the dose as the study progresses?
It's always great to get a question from Ben. And so absolutely. So not only as, again, in this study, but also for our future studies, we'll continue to dose escalate and build in the appropriate safety checkpoints along the way as well.
Claudia asks 2 questions back to back. Let me see. How long would it take for the FDA to approve the drug once your research has been finalized? And then, Dr. Christine, are you taking any new patients?
Yes, I am. I'll let
Okay.
Eric.
Yeah, not for me to answer Dr. K's schedule, but certainly, look, with respect to how long would it take the FDA to approve, our hope is to be within and conducting a study in the US in the next 18 months or so, and our hope is that given it's an orphan disease, that could be a pivotal or registration study. And so, when we think about being on market and the time that it takes, it's probably into the late 20s, 27, 28, in that range. But as an orphan disease, that does bring with it, and we do have an orphan designation for this specific molecule already with the agency. There is a drive and an impetus to bring therapies, for choroideremia patients, for RP, for all these types of patients that are affected. So we will continue to push in the appropriate way.
Are you taking patients? I'm thinking you said yes.
I'm in Gainesville, Florida. Vitreoretinal Associates, and you can Google the phone number. But yeah, I mean, it's I'm open to new patients. So
perfect. Here's the answer we were looking for. Conan Technology is a company that develops objective perimetry. There we go. Eric,
I'm not familiar with it, but the one of the challenges with perimetry is that with patients, they have to have the ability to fixate. And so, that's why older school approaches like Goldman kinetic perimetry are still appropriate. But, thank you. And I'll certainly take a look at it.
Our own Eric is asking, does your drug have any movement in the optic schism? Is that, I'm pronouncing that correctly? The nasal retinal fibers from each optic nerve something
Becky
crossovers
said,
into the.
yeah, into
Eric,
the.
you're
So
asking such hard questions.
Well, I can see the question here. I brought it up, so I'll answer it.
Okay.
The long and short of it, Eric, is that we don't know. And so you know, if one were to observe some contralateral effects, I wouldn't be surprised. Now, whether the drug is actually physically migrating through the chiasm and into the other side, if you will, or it's dampening, because these drugs are known to stabilize membranes, and so it could be dampening noise. At the end of the day, vision, native vision, is a binocular activity, and the brain is interpreting binocular input. And so it's a complicated question, but the definitive answer is I don't know whether the drug itself moves through the chiasm.
Bob has two questions back to back. The first, what does this treatment do to working photoreceptors? Meaning it creates new connections, but does it affect working PRs? We'll do one at a time. How about that one first?
What does this treatment do to working photoreceptors? Meaning it creates new. So really, Bob, I would say that it doesn't do anything to working photoreceptors. So as I mentioned earlier, with respect to affecting native or wild type retina, you don't see uptake of the drug in retinal ganglion cells, which sit downstream from normal photoreceptors. So photoreceptors that are retaining native function, they're sending their signals through amacrine cells and bipolar cells and getting to RGCs, and we're really not seeing the drug in normal RGCs, or RGCs rather, that sit downstream from normal receptors. Where we make an effect is that those that sit down from degenerated photoreceptors, we are making that retinal ganglion cell photoreactive or photoresponsive. So, and does that affect those photoreceptors that sit upstream? Don't know, but I suspect not. And I see the second question: what, if any, concerns would there be if treating someone who's already had a subretinal gene therapy? You know, look, it's tough. I mean, there is a you know, gene therapies have kind of a, I don't think they're definitively this way. And Dr. K, I'd welcome your input on this, but they're traditionally sort of a one and done type approach. Whereas our drug as a small molecule is titratable. I mean, it will be by definition something that gets repeated. What we will end up doing is likely working with sustained release technologies so that we can decrease the frequency of those injections and then have the drug slowly leach out, if you will, with a particular type of kinetic profile. And does it mean that someone who's had a gene therapy would be not applicable for this kind of therapeutic approach? No, I don't think so. In a study, it can be difficult as well, because it becomes confounding, because you've already had someone who's had one intervention. And so it's really tough to kind of tease out sometimes in clinical studies. So, the rules of clinical studies are different at some level than reality. So there's kind of two questions, or two answers to that.
Right, I agree. I mean, I don't think there's any contraindication medically or safety wise of doing a small molecule therapy after a subretinal gene therapy. I think that would be safe and fine. However, like you pointed out, it would be typical for that patient not to be a candidate for a clinical trial if this is still in clinical trial stage, because it's going to be in the exclusion criteria that they had another invasive treatment. And that's across the board for all clinical trials. That's quite standard.
Mm-hmm. We've heard that before. Steve asks, is there a timetable for clinical trials in the US? If so, is there a thought on how many sites, patients to begin with?
Yeah, the timetable, and you can see this on our a little bit about it on our website, is that 12 to 18 month timeline. I can't speak to exactly how, it's premature to speak to how many sites or patients, and what that might look like. But look, we would want a representative sample of subjects, and I think we would be going to the classic, if you will, centers of IRD, excellence and expertise, and including Dr. K's clinic and others. So I don't think it would follow or be that different from other studies that have been brought to the U.S. And that is absolutely our intention in our next step once we do this follow-up study within Australia. And I see kind of as a tail the next question, the race percentages of testing patients in age groups. There are no exclusions based on demographics per se. I mean, we're getting into the weeds a little bit. Some of these functional tests do require mobility. And so, we want to make sure that people have enough mobility and be able to physically carry out some of these assessments. We do typically, we shy away from syndromic types of conditions where there can be confounding and lots of other things that are affecting subjects. But I would say generally, the answer to your question is no. There's not a hard stop per se.
I think we have 5 questions left, and then we'll be wrapping it up. We still have most of our people with us, so we'll keep going. Is there any physical evidence of efficacy, meaning other than visual fields, obstacle courses, etc.?
Well, those are good physical evidences or objective evidence for efficacy. And so, we kind of think about buckets of efficacy. I'm assuming you're asking for this particular study, but that may be a generic question where you have, and Dr. K spoke about this, the agency, the Food and Drug Administration, likes to see, changes in visual acuity, and they like to see changes in functional testing. And they have traditionally also accepted changes in visual field. So these are the clinically, meaningful endpoints. Now, how you measure them and what constitutes a win is still a point of healthy debate and discussion with the agency. But I don't think that is going to necessarily change in the next 12 to 18 months. I mean, the general categories. I don't know, Dr. K, if you want to.
Well, I think it's an interesting question.
That.
You bring up a good point. I mean, I guess what you're asking for maybe is a structural evidence. So the FDA, we think about buckets of endpoints. So we think about visual function. That's things like perimetry and visual acuity. FDA clearly likes that and approves that. We think about functional vision. Those are mobility mazes, tabletop tests. FDA clearly likes that. And then we think about structural endpoints, and you could call that objective because you can't change what your OCT measurement thickness looks like. You can't have a placebo effect. So in a way, that's a separate bucket. But interestingly, the FDA doesn't necessarily like that any better. In fact, the only structural endpoint that has been sort of accepted by Wiley Chambers and the FDA that I'm aware of in inherited retinal disease that's regarding the OCT is something called the ellipsoid zone, which is the line that is the surrogate for the photoreceptor layer because it's a surrogate for visual acuity. And so, for instance, a very objective thing you could think about like electrophysiology. Let's put an electrode on your eye and measure how the cells are firing. The FDA's clearly told us all multiple times that they really don't care and they don't respect the fact, a change in an objective measurement like electrophysiology because it doesn't answer the so what question, which is what the FDA really wants to know. Has it impacted the patient's life? Are they functioning better? And that's what we want, a clinically significant and meaningful endpoint. So I think I would echo what Eric's saying here and not discount the things that you sort of just mentioned. Those are very important. In fact, those are the most respectable things for the FDA for an endpoint because those are impactful to a patient's function, the visual acuity, the perimetry, things like that. And in a way, structure is interesting. We absolutely still follow it, but it's sometimes even harder to get that to argue that actually is impacting a patient's life.
Good response. Carol asks, would this therapy make other therapies still viable when they become available? That's
I mean,
fantastic.
I don't think that it would have a clash at first. I think it would depend on what the other therapy is available. I mean, in the context of this conversation, again, in an outside of a clinical trial setting, if someone had a gene therapy, would that make this small molecule approach contraindicated? And I think the answer to that question would be no. That would not be the case. And so, in fact, you could even use it as a potential, this approach as a screen to see whether a particular, genetic manipulation could be viable and whether someone would benefit from it. So I do think there's absolutely some complementary components to this. I see the next question. When do we anticipate announcing the results of the Abacus trial? In the fourth quarter of this year.
And then 2 more questions. What are the thoughts of treating non-symptomatic carriers? Good question.
I'll let maybe Dr. K take that approach. I mean,
I mean, are we asking for this therapy?
Yeah, I
I
think for
guess
this
so.
particular
Mhm.
therapy, I'm not sure that it would be indicated.
No.
If you just had someone with really no, subclinical disease, I'm not sure that this approach would make too much sense for those folks.
Correct. Yeah, I think you have to have visual dysfunction to warrant this therapy.
We have one more here again from Susie. I think it was on slide 18, but it did show that on day 3, there was more brain activity on day 3 in a cluster area than the later days. Any insight as to the changes?
Yeah, I mean, look, fMRI is a fascinating research tool, and one that allowed us to say, does the brain light up? And by no means do I mean that it's squishy. I think when you look at the timeline, what we're seeing is that we do see the kinds of benefits that you're seeing throughout a month period of time. Now, the difference is that it is a small molecule, so it will have kinetics associated with it, meaning that it will get metabolized and it will get excreted. And so as we learn more about that, we'll be able to better answer that. At the same time, the drug is known to sit on the retina and so provide benefit. So I don't read too much into the kinetics of the MR, but at the same time, it does certainly support longer than just a day 2 or day 3 activity, and the other areas of evidence also support that.
I think we have one more question that is important, and then we'll end it. Promise, everybody. It's from Claudia. I understand that this is all very new, but would this treatment be affordable for the population?
We have an obligation and duty. I mean, we would look, and I don't think it would be really any different than approaches to treat wet AMD that are currently on the market. I will point out that as a small molecule, we are talking about a completely different economic argument relative to a gene therapy. And again, I'm not poo-pooing gene therapy approaches at all. There is a absolute role for them, and like I said, we I think complement them. But the costs associated with gene therapy approaches, we play sort of a different sport, and it's at a completely different level. So in short, I think the answer would be yes. It would be affordable.
That's it, guys. I'll just tell you too that this is the largest attendance I think we've ever had.
Well,
It's pretty exciting. I think it generates a lot of encouragement, and certainly for our viewers, as we said, the SAB is reviewing all of the work right now, and just keep your eyes peeled and email, and things will come out from the foundation. Certainly, thank yous are starting to come in on the chat. We certainly want to thank everybody, especially for hanging in there, Dr. K.
I know, with no power and on a generator, that's at least a couple beignets. So.
Actually, my husband texting me that we are on which is a terrible time to lose power because everybody else in the state of Florida has no power. And so even though we probably have like some tree limb down, we might be without power for days, is what my husband just texted me. So I'm like, oh my God. And then we run out of propane because I have a generator that runs off propane. So wish me luck on that, guys.
Good luck,
Anyway,
Dr. K. Good luck.
wonderful. Well, take care. Have a good night.
Thank you
Thank
so
you.
much.
Thank you.
And to
Thank you,
everybody, and thank
everyone.
you, Eric. Thank you for coming to and joining us. And we look forward to learning more as the days go on. Have a good night, everybody, and we'll see you next
My
time.
pleasure. Thank you.
Good night.
Bye-bye.
Good night.